Welcome to the lecture on dietary fuels and metabolic pathways. This starts the second half of the course where we're going to deal with intermediate metabolism, which is a big deal for biochemistry. Now there's some required reading here that's set out by these page numbers, and if you have the textbook or the online textbook, it's really important to read these. These are the big picture summaries that tell you how metabolism works, what the goals are, how the body works together. There's a supplemental file called Lecture 26 Reading that tells you how to find these particular pages if you have the online textbook. Sometimes it's not immediately easy how to see that, but you really do need to read these. It's very, very important. Again, as always, the lecture objectives are going to be important. It tells you the terms you need to learn. It tells you a little bit about what we're talking about. So I always say the great place to start for study is the lecture objectives, and that is very true here. When we say metabolism, we're talking about the sum of all the enzyme catalyzed reactions that occur in a living organism. If you've had biochemistry before, you know that we're actually talking about intermediary metabolism, which is all the enzyme catalyzed reactions that take place inside a cell. And that's more what we'll be talking about. We'll be talking about liver cells and muscle cells and fat cells, those kinds of things. But the key point is metabolism is a very dynamic process. It's always changing. It's very coordinated. It's very regulated activity, and it's always changing based on the needs of the individual. So balance is going to be a key point here. And we see over here the idea. The idea is that as living beings, we have to expend energy to do things like work or synthesize or store. And that work is going to take energy. Where does that energy come from? It comes from the fuel molecules that we consume in our diet. We consume carbohydrates and fats and proteins. That gets digested, broken down, absorbed, and then the intermediary metabolism pathways that we have will do a series of chemical reactions on those molecules to extract their energy and to convert that energy into a form that we can use to do the things that we need to do. Sometimes we will do the work of living. Sometimes we will store excess energy in other fuel molecules. In the end, we will have waste products, CO2, we breathe out, and there are the waste products that we will eliminate. And that brings up the idea of a metabolite, which is a small molecule involved in the synthesis and degradation of other molecules or biopolymers like proteins or oligosaccharides or whatever. And that's going to be all of our fuel molecules that we consume. We're also talking about vitamins and cofactors because they're involved in our enzymatic reactions, as we saw before, and any intermediate molecule in any of our pathways. And we'll see coming up examples of allosteric modulators as being regulators for these pathways as well. So bringing up the term metabolic pathway, what does that really mean? Well, it's pretty obvious, as you would imagine. A series of coupled reactions in which the product of one enzyme reaction is going to be the substrate for the next enzyme reaction. And it also implies if it's a pathway, there's going to be some regulation. So for example, glycolysis is a pathway. We have a whole lecture on glycolysis that we'll talk about later, where we take glucose that we've consumed and it will go through a series of chemical reactions that will transform it structurally and energetically. And as we move along from one reaction to the next, we find our way down here to the product pyruvate. So it's a pathway in the sense that there's a 1, 2, 3, 4, 5 order to the pathway, and the product of one enzyme becomes the substrate for the next enzyme. So don't worry so much about the actual pathway. Like I said, we have a lecture coming up. And what's interesting is there's also a pathway called gluconeogenesis, which is the pathway in our liver where we can synthesize glucose from starting materials. And largely it's going to be the reverse of glycolysis, using many of the same enzymes, a couple of differences that allow it to be regulated differently and allow the energetics to go differently. But the key point for both of these pathways is there are specific enzymes at key points in the pathway that are regulated to control the flow of metabolites through the pathway. And that's what these boxes are telling you. If you want to go to glycolysis, you want these enzymes to be fully active and fully operational. If you do not want to go to glucose, you want to turn off these key enzymes, make them inactive. Or if you want gluconeogenesis to work, these enzymes will be active and you want the glycolytic enzymes to be off. We'll see that coming up here. And let's review our dietary fuels. We will have a course on nutrition in this program, and you'll really get the nutrition details, digestion, absorption, calories, all that kind of stuff. But for biochemistry, let's look at the basics. For carbohydrates, we know there are 4 kilocalories of energy stored per gram of carbohydrate. That's not so bad. Typical carbohydrates that we consume are starch, which is found in vegetables, plants. Sucrose is table sugar, lactose is milk sugar, fructose in vegetables and fruits. Of course, glucose is a sugar. It shows up a lot of places. And we know that there are no essential carbohydrates, which means there's not a single carbohydrate that we have to consume to survive. Our body can make whatever carbohydrates we need. We'll cover that coming up. Proteins, amino acids, they also have 4 kilocalories per gram of protein. And that's any polypeptide that's composed of amino acids. But we do know there are 9 essential amino acids. We saw that in the amino acid lecture, and they're listed here. And those are the ones that our bodies cannot synthesize, so we must get those in our diets. Fats, 9 kilocalories per gram. That's a tremendous amount of energy. That's why our body likes fat so much. That's why our body craves it in taste and wanting fat, and that's why our body stores fat. It's very, very efficient. And the fats we consume are primarily triacylglycerols or triglycerides, or we also get fatty acids and lipids. You get them from plants, you get them from animals. Fats are everywhere. And there are essential fats as well. Linoleic acid and alpha-linolenic acid are essential fats. We mentioned those back in the fatty acid lecture. And believe it or not, alcohol, ethanol, is a dietary fuel. It's not at all required. We live just fine without alcohol. I'm not saying for 1 second you need to consume it as a fuel, but when alcohol goes into the body can metabolize that alcohol and get 7 kilocalories of energy for every gram of ethanol. And never forget that we need vitamins, minerals, and water. Another term is catabolism. Catabolism, or to catabolize, is to break down biomolecules in order to produce energy. And in metabolic terms, we're doing that via oxidation. And oxidation means taking electrons away from the molecule. So if you add an electron in, you're reducing it. If you pull an electron out, you are oxidizing it. So primarily we'll be removing electrons. Now, if you know organic chemistry, you know sometimes an oxygen atom will show up as a result of an oxidation reaction, but make sure you keep it clear that oxidation is electrons and reduction is electrons moving in, oxidation electrons moving out. So they produce energy and they produce building blocks for other synthesis. And the fuels are always going to be oxidized completely to CO2, which is what we exhale, and then, of course, water. And what's interesting about the catabolic pathways is they all converge on just a few products. For example, if we consume carbohydrates, if we consume glucose, that pathway of glycolysis is going to lead us to pyruvate. Pyruvate takes us to acetyl-CoA. If we consume fat, the fatty acids are also going to be oxidized. We're going to have some energy molecules are produced, and the product is acetyl-CoA. And for amino acids, when we oxidize amino acids, we also produce acetyl-CoA or sometimes other intermediates as well. So then we say that the acetyl-CoA is a common intermediate for these catabolic pathways. Now, the energy that is released by catabolism, as you mentioned, pulling these electrons out, pulling the energy out, you don't want to lose that energy. You want to capture that energy. And so the energy that is released by these catabolic pathways is going to be conserved in 3 types of molecules. We have nucleotide triphosphates, nucleotides, you know, familiar with ATP, GTP, CTP, that kind of thing. Those phosphate bonds contain energy. The other one is going to be these reduced coenzymes. We saw that in the vitamin lecture, NAD, NADH, or FAD, FADH2, these kinds of things. These will hold those electrons. They will hold that energy, and they will move that energy from one system to the next. And then acetyl coenzyme A, or acetyl-CoA, is also an important molecule bridging different pathways. If you can tear something down, you can build it up. So anabolism or anabolic is going to be the biosynthesis of more complex molecules from smaller precursors in reductive pathways. For catabolism, you're breaking down a molecule and you're oxidizing it. You're pulling the electrons out. For anabolism, you're actually synthesizing or making new molecules, so you're investing the energy, you're investing electrons into that molecule. That's why it's a reductive pathway. And so for anabolism, you're investing energy into a molecule so that molecule can be stored, or that molecule can be released to the blood so that other tissues can use it. So you put energy in to make these molecules, and that energy will be carried by that molecule to be stored or used elsewhere. Anabolic pathways are divergent, starting with a few metabolites, and they can produce a whole lot of different things. So we can form glucose starting with glycerol, we can form glucose starting with alanine, we can make glucose starting with lactate. So 3 different molecules you can start with, but as soon as you get into the gluconeogenesis pathway, pretty soon you're on a common track, and they all lead to the final product, which is glucose. Here's a direct comparison of a catabolic pathway, glycolysis, with an anabolic pathway, gluconeogenesis. Now, just as a reminder, we have a whole lecture on glycolysis coming up and a whole lecture on gluconeogenesis, so don't focus on the details right now. But when you look at a pathway and you see a fuel molecule like glucose or fatty acid or amino acid or alcohol, and you see a series of reactions that result in electrons being extracted or ATP being produced, you can be pretty sure that's catabolic. You're doing a metabolic pathway designed to take energy out of the fuel molecule, transform it, and then use it in some way to produce ATP. Over here, for an anabolic reaction, you see some starting materials that are not what we would consider fuel molecules or something the body could use, and you see a series of reactions that will invest energy, like here, investing electrons or investing phosphate, high energy phosphates, and you're heading back towards the formation of a fuel molecule that can be used by the body in some form. That is going to be an anabolic reaction. The next terms we look at are aerobic versus anaerobic metabolism, and those are fairly common terms, aerobic with oxygen, anaerobic without oxygen. And for metabolism, we're thinking about ATP production. And so this is ATP production that depends on molecular oxygen versus ATP that does not depend on molecular oxygen. And so we have anaerobic ATP production and aerobic ATP production. And to understand this, let's look at the figure here. Now, just as a reminder, we have a lecture on glycolysis, we have a lecture on fatty acid oxidation, we have several lectures on amino acid reactions. We have a lecture on the TCA cycle, we have a lecture on the electron transport chain, and we have a lecture on the ATP synthase. And at all times, we'll be working these things together, back and forth through all these pathways, and you'll have plenty of opportunity to learn these pathways and how they work together. So for now, please focus on the general picture and the idea, and don't get pulled into the details quite yet. So if we look here, the electron transport chain is going to receive electrons that are removed from fuel molecules, glucose, fatty acid, amino acids. These will be oxidized, and those electrons will be removed, and those electrons will be sent down here to the electron transport chain, where they will move through the chain, and they will lead to the production of ATP by the ATP synthase. If you'll notice here, there is an oxygen. That molecular oxygen is part of the electron transport chain. It absolutely must be there for the electron transport chain to work. So in order for the ATP synthase to make ATP using the electron transport chain as a driving force, then molecular oxygen is required. So this is aerobic ATP production. So what does that mean then? It means that fatty acids are going to be producing electrons, and amino acids are going to be producing electrons, and heck, even glucose is going to be producing electrons that will go down to the electron transport chain. So in that regard, these processes here are going to be aerobic ATP production. The TCA cycle is going to be a set of reactions that take place inside the mitochondrial matrix, and they're going to be extracting electrons, and they're going to feed the electron transport chain too. So we see how that works together. So anytime you see NADH or FADH2, these are reduced cofactors that are carrying electrons to the electron transport chain. And sure enough, you see glucose, glycolysis will make these molecules as well. But glucose to pyruvate, glycolysis is a little bit different. Here, you see the production of ATP occurring, and there is no molecular oxygen taking place in the glycolysis pathway here. And because of that, we say that glycolysis can produce ATP in the absence of molecular oxygen. Therefore, it is anaerobic ATP production. Yes, of course, glycolysis does make these reduced cofactors or lead to the production of cofactors, and these can go to the mitochondria and they can produce so much more ATP. But glycolysis is the one metabolic pathway we have where you can produce ATP in the absence of molecular oxygen. So how does this all fit together then? Isn't glycolysis dependent on oxygen? Well, kind of. As I mentioned before, part of glycolysis is to pull electrons out of molecules and to put these onto these cofactors, NADH here, they can carry this down to the electron transport chain. And there's FADH too. And that's what fatty acid oxidation does, and that's what amino acid oxidation does. You get these reduced cofactors and they carry the electrons to the electron transport chain. So while glycolysis can produce ATP in the absence of oxygen, if you have mitochondria, if you have molecular oxygen, those electrons that are being carried away from this pathway can go down and produce more ATP. And so all of our energy production in our cells that have mitochondria is going to be substantially increased by oxygen-dependent ATP production or aerobic ATP production. So what would happen if you go ischemic? What would happen in the brain if you have a blockage, which could be stroke, or in the heart you have a blockage, could lead to a heart attack? When oxygen is low and tissues don't have enough oxygen, the electron transport chain will not function very well. If it doesn't function very well, that means it's not going to be using the electrons that are being sent to it from these other pathways. If the electron transport chain doesn't use those electrons, then the concentration or the amount of that reduced cofactor in the cell is going to get bigger and bigger and bigger and bigger. It's not being consumed because there's not enough oxygen being driving this electron transport chain. And so we'll see coming up pretty quickly that the NADH produced by the TCA cycle is a very strong regulator of the TCA cycle. And the NADH produced by glycolysis and by fatty acid oxidation and through amino acids is also a pretty strong regulator. When a cell goes ischemic and the NADH levels start to rise, all of our energy production pathways will become inhibited and they will function at a much lower rate. And so glycolysis is the only pathway that can continue to make ATP in the absence of oxygen or in a low oxygen environment. But even glycolysis will be affected. If the oxygen remains low for a long time and these NADH levels rise, the NADH will also start to slow down this glycolytic pathway. And we're going to talk quite a bit about that coming up, especially in the glycolysis lecture, but then in other lectures as well. And so the rule of thumb is glycolysis does allow for anaerobic ATP production, but in a cell with mitochondria, the products of the glycolysis pathway goes on to do aerobic ATP production, and that can be affected by oxygen levels in the cell. One of the most important points we have for metabolism is the idea of balance. Cells will always work to balance. The cell will say, do I have energy now? Do I need to make energy? Do I have excess energy? Do I need to use a fuel molecule? Do I need to store a fuel molecule? All of these things are going to be important questions that our cells and our organs and our systems will be asking. So here's an example of the TCA cycle, and remember we have a lecture coming up on the TCA cycle. And the TCA cycle is designed to oxidize molecules, use those electrons in the reduced cofactors to support aerobic ATP production. So during times when the cell needs energy or is a little bit low on ATP, the TCA cycle will be working at a very high level to help produce ATP. But what happens when a cell is more relaxed? When a cell says, I've got energy, there's not a lot of demands on me right now, I really don't need to have a lot of TCA cycle activity. What can I do with this very powerful TCA cycle that I have? Well, you'll notice that there are molecules in the TCA cycle, and those molecules, if they're not needed for the TCA cycle, they can leave the cycle. They can go off and do other things. You can synthesize fats, or you can synthesize amino acids, or you can make neurotransmitters, or you can make heme, depending on what cells you're in. Or if you're in a fasting state, You could say, well, I'm going to make glucose so I can send glucose out to the blood or whatever. And so these are examples of how the cell says, if I need my TCA cycle, I'm going to use these molecules for the TCA cycle. But if I don't need the TCA cycle to be so active, I can do other reactions. And we're going to see how that balance works in a very, very nice concerted effort. And this is very true for the cell overall. This is one of my favorite pictures. And if you've had metabolism before, you look at this and you say, got it. If you haven't had metabolism before, you look at this and say, am I ever going to get this? And we will. As we move through our carbohydrate metabolism, as we move through our fatty acid metabolism, all of this stuff will make a lot of sense to you. But what you'll notice here is that this is telling you it's a liver cell. It's just a single cell within a liver. I like the way they do this. We know there's a lot of different types of liver cells, but this is just sort of your general. And you see glucose can come in, and glucose can go into the glycolysis pathway, as we've talked about before. It's also possible for fatty acids to come in, and fatty acids can get into the mitochondria, and they can do fatty acid oxidation. We have amino acid processes going on. And so the other thing the liver can do is it can say during fasting, it's going to say my job is to produce glucose for the blood. And so instead of doing glycolysis and using glucose as an energy source, the liver will flip over and say, I will start synthesizing glucose and I will release that glucose into the blood so that other organs can have it. And so the question is, what is the balance? How does the liver know it's time to use glucose for energy? How does the liver know it's time to make glucose to be sent back out? And the same thing is true over here for fats. How does the liver know whether it's time to bring fats in to use them as energy, or whether it's time to synthesize fats and send those fats out into the blood? And the balance is going to be the key. And the balance is going to come from what our metabolic state is at any given time. Whether we've consumed a meal, whether we have fresh foodstuffs that are entering our blood and we can use those for energy, or whether we're fasting and we need to use available fuel resources to try to meet our needs. And the liver is going to be very sensitive to those signals. And then you generalize this to the whole body. And so in this case, we'll see coming up a couple more slides from now, if a person has just consumed a meal, they have carbohydrate, fat, and protein in their gut, and these molecules are being absorbed. And so we see glucose is in the blood, we see fat in the blood, we see amino acids in the blood. And the question is, who cares? Well, every tissue loves glucose. We'll see this. The liver, the brain, red blood cells, skeletal muscle, adipose tissue, all peripheral tissues, everybody loves glucose. And some tissues love fat. Adipose loves fat to store it. Muscles like fat because they use it. And so the balance is going to be dependent on whether there's an excess of fuel molecules or whether there's not. So this is an example of the fed state when these molecules are available from our diet and they're being absorbed, and all of the tissues can make decisions based on what they like best and what they want to do. We'll see coming up in a few more slides that during fasting, when there is not fresh fuel molecules coming in from the gut, it's going to be up to these organs to share with each other and provide for each other based on their own metabolic needs. And it's all going to be based on that same metabolic state that we're in. So 2 slides ago, we talked about the idea of the liver making a decision whether it would want to do glycolysis and then use glucose for its own energy needs, or whether it would want to synthesize glucose in order to release that glucose out in the blood. And if you recall, those 2 pathways here, here's the glycolytic pathway and here's the gluconeogenesis pathway. And they're really very similar in this part. Of course, the arrows for glycolysis are taking glucose in a catabolic direction. Over here, we see glucose is being synthesized as part of an anabolic reaction. But you'll notice that these steps are almost identical. And if the liver were to do both of these at the same time, that would be problematic. I mean, right away you can say, if you're a liver and you're using glucose as an energy source, why in the world would you want to synthesize glucose if you're going to be burning it for oxidizing it for energy? Or if you're in a state where your job is to synthesize glucose and you're doing all that effort to synthesize that molecule and produce it, why in the world would you want to turn around and immediately oxidize that glucose? And so that sets up the idea of a substrate cycle. A substrate cycle is where glucose can come into the liver and be oxidized and make all these products. But under different conditions, the liver would make that glucose molecule. And so those same product molecules from glycolysis can be the starting material to make glucose by the liver. So that's just the normal life cycle of how molecules move and go from the normal. But it also brings up the idea of a futile cycle. And the futile cycle is right here. For glycolysis, again, we'll see the detail when we do the pathway, but the first step of glycolysis, when glucose comes into the liver cell, the glucose is going to be phosphorylated using a molecule of ATP, and that phosphorylation will trap the glucose inside the cell, and then once it's trapped inside the cell, then the catabolic reactions can take place. If you're going to be synthesizing glucose, one of the last reactions you do is you take the phosphate off of that glucose. You dephosphorylate the glucose to make free glucose, and that is the free glucose that you release out into the blood. So if both of these pathways are active at the same time, if both of these enzymes are active at the same time, you could bring glucose into the cell, you could phosphorylate it, and then this phosphorylated glucose could find its way over here to this phosphatase, and the phosphatase would just pull that glucose off, and out it would go. And that's a futile cycle, meaning you consume a molecule of ATP to phosphorylate, and then you immediately dephosphorylate that molecule. You've wasted that ATP. There's nothing to it, and that's what you see here. Glucose could be phosphorylated, glucose could be dephosphorylated. That would be futile. That would be wasting energy. Well, how do we prevent this? What does the cell do to prevent this? Well, the cell will make sure that under times when the liver wants to use glucose as an energy source, then the glycolytic enzymes will be active. All of these enzymes will be active and they'll be on and they'll be functional. During those times, several of the enzymes in the gluconeogenesis pathway will be shut down. They will be negatively allosterically modulated, so they have very little activity. And that makes sure the gluconeogenesis pathway is off at the time that the glycolytic pathway is on. And then you can imagine during times of fasting when the liver is supposed to make glucose and send glucose out to the blood, you want to make sure the glycolytic pathway enzymes are shut down, and you want to make sure the gluconeogenesis pathway enzymes are turned on. And that's what we see over here. This is showing you that the blue-green arrow is showing you that you're trying to move from glucose 6-phosphate towards glucose, and that's fine. You could do that with the glucose 6-phosphatase to make that glucose. And then to make sure that this glucose is not immediately rephosphorylated, this little block here is showing you that the glucokinase, this enzyme right there, is going to be inactivated during times when glucose 6-phosphatase is active. And that's called reciprocal regulation. Reciprocal means if I have two pathways that are paired, one is off, one is on, and then at other times the on turns off and what was off turns back on. And the way that works in the liver is kind of interesting. If we start over here on the right, if you look at this glucose 6-phosphate, however that glucose 6-phosphate is produced, if it's produced by gluconeogenesis, if it's synthesized, which you see right there, we want to make sure that we dephosphorylate this glucose, and we want to make sure we protect that glucose from getting rephosphorylated. So during times of fasting, when the liver is synthesizing glucose, there's a specific protein in the endoplasmic reticulum membrane that will bind that glucose 6-phosphate, and this glucose 6-phosphate goes inside the endoplasmic reticulum, and that takes it away from the kinase enzyme that would rephosphorylate it. And you see here that the dephosphorylase is there. You take the phosphate off of the glucose is allowed to exit out of the endoplasmic reticulum, and while it shows a big distance here, this glucose is then routed out to the plasma membrane so it can be released. And so this is an example of compartmentation, where you do this dephosphorylation reaction in a different compartment than where the phosphorylation takes place, which is the cell cytoplasm. Now, if you're thinking about glycolysis, when the liver says, I'm going to use glucose as energy source, and I'm going to oxidize them and do the catabolism, under conditions where the cell is using glucose, this transport enzyme is going to be turned off. So if you bring a glucose in and you phosphorylate it, you want to make sure that it does not get transported into the ER to be dephosphorylated. So you turn that transport enzyme off, and then the glucose 6-phosphate can go forward. Another really good example that we'll develop later is when we do fatty acid oxidation. When fatty acids come into the cell cytoplasm, they're brought to the mitochondria, and they need to be put inside the mitochondrial matrix because that's where fatty acid oxidation takes place. And there's a very important enzyme here called the carnitine palmitoyl transferase 1 enzyme. don't memorize that for now. We'll talk about it later. But it will take a fatty acid and it sticks a molecule of carnitine on that fatty acid instead. And that carnitine is like an ID card. It's like a pass card that allows this transporter to move the fat into the mitochondria. This transporter will not move the fat unless there's a carnitine on it. And then once the fat gets on the inside here, the carnitine comes off and you have the fatty acid inside. Okay, that's all well and good. Why is that important? There will be times that the liver cell will synthesize fatty acids. It takes excess energy. When it has too much energy, it'll synthesize fats because it can store the fat very efficiently. So here we have the fatty acid synthesis pathway taking place, and you'll notice that you produce fatty acids. And these fatty acids are supposed to be released from the cell so they can go to adipose and be stored. But what's going to stop this newly formed fatty acid from coming over here and binding to that carnitine palmitoyl transferase 1 and getting a carnitine on it and for it to get shoved into the mitochondrial matrix just to get oxidized? That would be a futile cycle. It turns out that during fatty acid synthesis, one of the very first molecules that's made is this molecule melanoCoA. And melanoCoA is a very strong negative allosteric modulator of that CPT1 enzyme. So when the cell gets the message that it's time to start synthesizing fat, melanoCoA levels start to rise because this pathway is active. And very quickly, the CPT1 enzyme is completely shut down and no fat can move into the mitochondria. Okay, so after the fat synthesis process is done, when the liver is done making fat and it gets the message it's time to stop synthesizing fat, it's time to start using fatty acids as energy, then we'll see the fatty acid pathway will stop, melanoCoA levels will go down because it gets consumed, and then eventually the melanoCoA level is very, very low. And when you know the inhibitor is low, then the enzyme is no longer inhibited, and that's going to allow this CPT1 enzyme to become active again, and it can start transporting fats in. So again, another example of compartmentation for regulation. So when you think about biochemistry, there is a physiology component where we consume foods and it gets into our gut and all the stuff that happens in the gut to absorb those fuel molecules into our system, and that'll be covered mostly in the physiology class. And then you think about, when we say fat, what do we really mean? Do we talk about saturated fat, unsaturated, polyunsaturated? Do we talk about, essential amino acids or just regular protein? Are we talking about dietary sugars? That'll be covered in the nutrition class. So we'll talk primarily about carbohydrates, and we're thinking glucose, even though we know there's fructose and galactose in our diet, and amino acids in general. So we're going to pick the story up once these molecules get into the bloodstream and once they reach their target place. And the overall rule always is, whatever tissue you are, muscle, brain, red blood cells, liver, you're going to have different needs for different fuels. And the first question is, any fuel that gets consumed by any tissue, you're going to use that fuel to meet the immediate energy needs. That tissue is going to say, I'm going to use this fuel to meet my own needs first. And once those energy needs are met, once the cell feels like it's got plenty of energy, then it starts to make a decision. What do I do with this excess energy? Do I want to store these fuel molecules? Do I want to modify these fuel molecules? And that's what we're going to be looking at. So glucose is the universal fuel. Every tissue we have uses glucose, and it can produce ATP by glycolysis. If you're a cell that has mitochondria, then the products of glycolysis can be used to make ATP aerobically. And so glucose can be used to meet immediate energy needs. But if you're a liver, then you're saying, I'm very interested in storing glucose for times of fasting, or I can convert the glucose into fats for storage at adipose. And so glucose is a pretty powerful molecule for the liver to decide what to do with. Amino acids, a little bit different. When amino acids come in, most tissues are saying, I need amino acids because I need to synthesize protein. If you're a muscle cell, you've got a tremendous amount of protein and a lot of protein synthesis activity going on all the time. And so that's going to be the primary use of those amino acids. But you could also be a nerve cell, brain, these kinds of things, and you're thinking, I need to make neurotransmitters. Or if you're a liver, you can say, well, I need to modify nitrogen because nitrogen is a molecule that is toxic if it's in an ammonia form. So I'm going to modify amino acids to take care of my nitrogen. Okay, and then fats, of course. Fats are great. If you're a muscle cell, you can use fat. If you're a liver cell, you can use fat, or even adipose cells will use fat as energy. That's fine. Red blood cells can't because they don't have mitochondria. But so red blood cells don't care about fat, but other organs do. So again, when fats come in, you can oxidize them to make energy, or you can store them if that's what you need. Every cell has membranes. Every cell needs lipids for the membranes, and so every cell is very interested in fat to help maintain the membranes they have. So that's the overall guiding principle of what is going to happen when our bodies absorb those dietary fuels. And of course, you'll see this will be for physiology, but also for nutrition. But when you're storing things, where do we store glucose? If we have excess glucose, it's going to be stored primarily as glycogen. Glycogen is a large polymer of glucose residues. We have a whole lecture on glycogen coming up. And the liver will store glycogen because that glycogen will be used to make individual glucose units that can be put out into the blood during fasting. Skeletal muscle, especially fast twitch skeletal muscle, likes to store glycogen because it needs a ready supply of glucose that it can use for ATP production during times of intensity and exertion. Fat is stored in adipose tissue, is where it's going to be stored. The liver can make fat. It wants to send it to the adipose. Adipose, can package it up and stuff. We'll see that. And then, of course, amino acids are going to be used primarily for protein synthesis. And we really don't talk about stored amino acids. There's really no place where you can say, I've got a bag of lysine or a bag of methionine or a bag of valine or whatever. Instead, amino acids are found in our blood and amino acids are found in our cells, and we call those the pools or the supplies, and they're readily available. And so our body will consume protein, break that protein down, it will hold those amino acid pools, and then if there's excess amino acids, a lot of times they're just eliminated through the urine or stool or whatever. Or the liver can say, I can turn an amino acid into fat just as well as I can turn glucose into fat. And so protein can lead to stored adipose tissue as well. And then one of the last big things we're going to talk about is the metabolic environment that helps these organs and these tissues make these decisions. And it's based on two fundamental metabolic hormones that we have. These are insulin and these are glucagon. And these two hormones are going to signal the metabolic state of the body. Now, insulin, we have a whole lecture on insulin and glucagon to talk about how they're produced and what are the factors that make them work the way they do. Insulin and glucagon are both going to be released by the pancreas, and the pancreas is going to pick up signals that's going to let it know whether it needs to release insulin or whether it needs to release glucagon. Insulin is going to be released from the pancreas in response to the presence of glucose in the blood, and that's going to be glucose coming from the diet. And we'll make a big deal about if a person is just at a regular fasting state, everything is normal, everything is healthy, there is a certain amount of glucose in the blood. We call that the resting blood glucose level. And when you're at that resting level, the pancreas says, I am where I need to be. I need to maintain that. And then when a person consumes carbohydrate, when they consume glucose, then it gets absorbed in the blood. The glucose level goes higher and higher and higher and higher. And that increased glucose will signal the pancreas that it needs to release insulin into the blood. And then insulin will circulate throughout the entire circulation. And any cell that's sensitive to insulin will bind that insulin. It will set off that insulin pathway we talked about in the signaling lecture, and that will stimulate these cells to bring the glucose out of the blood and into the cell. That way the cell can use the glucose for energy. What insulin is saying, I have a resting blood glucose level, and that's where I need to be. And I've seen a large increase in the glucose in the blood. And so insulin will tell the cells that glucose is available, and insulin will ask those cells to take that glucose up because I want to get the blood glucose back down where it belongs. So when the blood glucose is elevated, insulin is out. When the blood glucose is normal, the insulin is gone. So what about glucagon? Well, glucagon is just the opposite. Glucagon is going to say, I see a normal blood glucose level, and that's what I want. And if the blood glucose starts to decrease, if it gets lower and lower and lower, then the body will start to suffer. Brain needs glucose, red blood cells need glucose, and so we must maintain our resting blood glucose level. And glucagon is the hormone that tells organs in the system your blood glucose level is getting lower. You need to maintain a certain level of glucose production for the blood. And so we call glucagon the fasting hormone because it helps maintain blood glucose levels during fasting. And it doesn't just do blood glucose, but it's going to talk to different organs and it's going to say, you're fasting right now, you need to behave this way. And insulin will come along and say, hey, there's excess glucose. You need to behave this way. And you see these two are going to work together. They're going to work opposite to each other to control our metabolic state. And we're going to develop this quite a bit coming up. Okay, let's go through these metabolic states. And this is really the fundamental big picture for metabolism. So we have the metabolic states as being the fed state, which is going to be a period from when absorption starts to when absorption is completed. So what does that mean? All right, so you're sitting at the table and you've had a nice meal, you've eaten, you've swallowed, it's in your stomach, it goes through that, it gets into your small intestines and large intestines, and that material is starting to be absorbed. And as that absorption starts, those fuel molecules are now available to your body, and that is entering the fed state. And assuming a person eats a normal meal, takes, however long it takes to eat, half an hour, 15, 20, it depends. Okay, so all of that food stuff is going to move from the stomach to the intestines. Some of it's going to be absorbed, some of it's going to pass on through as waste. But at the time that the absorption is completed, that's when the fed state will end. And as you can imagine, as the food is first entering it very slowly starts to be absorbed, and when more food comes, there's more absorption, so it kind of rises to a height, and then it sort of falls off slowly as the food stuff. So there's no definite beginning and no definite end, but rather you enter the fed state and you leave the fed state. And as you leave the fed state, you enter the fasting state. And the fasting state is defined as that period that follows completion of absorption. When there's no more absorption going on, at that point, you say there's no more dietary fuels coming in. At this point, I have to rely on the dietary fuels that's contained within the body. I have to rely on the fat that's stored in the adipose. I have to rely on glucose that's stored in the liver. And we have to take care of ourselves. And then we have the third, it's called the long fast or the starve state here, which is when you're in the fasting state for more than 2 days. And this is interesting if you've had nutrition before. Different textbooks, different sources will say 48 hours, 72 hours. I'm not going to split a lot of hairs. I picked 2 days because that's what our textbook says. But I'll definitely listen to arguments about 2 or 3 days as well. I won't split hairs on this. But that's when we fast for longer than 48 hours. And our system will definitely take on characteristics when they say, boy, you haven't eaten for a couple of days. I don't see any fuel molecules coming in. I need to get myself set for a longer period of fasting. I need to conserve resources. I need to use materials a little bit differently. And so we're gonna adjust ourselves. And of course, if you're in the fasting state, as soon as you consume food, you re-enter the fed state. If you're in the starve state and you consume food, you immediately go into the fed state. So you see how these things will interplay and interchange. Okay, so here's the fed state. And this is a lot to look at and it's a lot to think about. But let's go over here to the right because this is the key. So our blood glucose level, it's called basal, our normal blood glucose level, about 80 to 100 milligrams of glucose per deciliter of blood. That's the range. Again, you can argue, well, we know that it should be lower than that or higher. Okay, 80 to 100 milligrams per deciliter is pretty normal. Okay. Now, here we are in the fed state. We've just consumed a meal. There's carbohydrate, there's fat, and there's protein. Let's assume that they're all there. So the first thing that happens is the carbohydrate gets absorbed, and that carbohydrate gets released into the blood, and that's going to cause the blood glucose level to rise. And as the blood glucose level rises above that 80 to 100 milligrams per deciliter, the pancreas will sense that increase, and it will know that it's time to release insulin. And because of that now, insulin will increase because blood glucose is released. Is increasing. And that insulin is going to circulate throughout the whole body. And any tissue that hears the insulin signal is going to respond to the insulin signal, and it's going to say glucose is available. I need to take this glucose in. So anytime you see this green plus on any of these tissues here, these are pathways that are stimulated by insulin. It's hard to see, but there's a little eye under that green plus. So let's look at liver. Liver is very insulin sensitive. When liver hears the insulin signal, it says, wow, there's a lot of glucose. I can bring glucose in and I can use that glucose for my own energy needs. I can do my own energy production, which is what this is showing you here. But then the liver says, well, I have more glucose than I need to meet my energy needs, so I'm going to take this excess and I'm going to build up my stored glucose, which is glycogen. And if there's even more glucose available, I'm going to start synthesizing fat because it's easier to store fat than it is to store glucose. So I'll start forming fats and triglycerides, and I'm going to release those so they can find their way and be stored down here in the adipose. All right. Well, let's see who else. Muscle, skeletal muscle. Skeletal muscle loves glucose because it's quick production of ATP. Skeletal muscle hears the insulin signal. Skeletal muscle will bring in the glucose and use it for its immediate energy needs. And skeletal muscle can also store glucose. And so the excess glucose over the immediate energy needs will be used to refill the glycogen levels here. So insulin will do that. And you see that adipose tissue is also sensitive to insulin. Adipose tissue loves glucose to use as an energy source, but it's also going to use that glycolytic pathway to make intermediates necessary to build and store triglyceride molecules. We have a giant lecture just on that here. Okay. All right, so if you're going to store fat, where's that fat coming from? Well, here's fat that's come in through the diet. And we'll talk about chylomicrons and how they get in. But anyway, this is fat that gets absorbed, and it's going to circulate through the body. Well, primarily, all of these organs are going to be very, very happy with the glucose that they're getting. They're just going to say, that glucose is more than enough for me. So you see, nobody really cares about the fats coming in. So that fat's going to go down here to the adipose tissue. And because insulin is stimulating glucose and stimulating triglyceride formation, those fats can be incorporated and stored into the adipose tissue here. Let's go over here. You see brain is a major customer of glucose. Red blood cells is a major customer of glucose. But you notice that neither one of these cell types is going to be sensitive to insulin. There's a very important reason for that. The big reason for red blood cells is that they don't have mitochondria. They cannot do aerobic ATP production. All they can do is do glycolysis and get their ATP anaerobically. So they're not going to say, insulin, you don't have to tell me to take glucose in. I'm going to take glucose in all the time, okay? And I'm just going to do it. So I don't need to hear the insulin signal. Brain is a little bit funny. Under most conditions, brain is going to want to use glucose as the main energy fuel molecule that it has. And so brain will suck up glucose out of the blood like crazy. And the brain will take glucose whether we're in a fed state. Brain will take in glucose when in the fasted state. So again, brain says, insulin, I don't need you to tell me to take glucose in. I'm going to take glucose in anyway because that's the way I'm supposed to be. All right, so what you see here now is that the insulin signal tells these tissues that have the ability to utilize excess glucose, it tells them to take in that glucose, and it turns on the pathways that will utilize that excess glucose, either for storing glycogen or for storing fat. That's the important difference versus brain and red blood cells that will take the glucose in just because that is the primary fuel that it needs. And of course, when you have protein, you have these amino acids, and every tissue is going to love amino acids to synthesize and stuff. When it says tissues here, it's talking about all peripheral tissues. So it would be muscle, adipose, every tissue that needs protein is going to do that. So here's the words, and these are really much the words that I've talked about, so we can go through this. But during fasting, when we don't have foodstuffs coming in, when we have not consumed a meal, our blood glucose is going to be maintained in the range of 80 to 100 milligrams per deciliter. And you could say, well, you know, people who are pre-diabetic have a higher. Of course they do. Or people who have, fine. But this is what's considered a fairly normal fasting blood glucose range. Okay, and so we're going to be thinking about the insulin to glucagon ratio. Okay, so glucagon is the fasting hormone. Glucagon is what changes our tissues to make sure the blood glucose stays at this level. And because you're at this level, there's really no need to release insulin because the blood glucose is normal. Insulin comes out when the blood glucose level gets higher than this. And so during fasting, you don't need insulin. During fasting, glucagon is maintaining this level right there. And so we call this ratio as low. It's low insulin, it's high glucagon. Glucagon is in control. But then if a person were to consume carbohydrates, were to consume glucose, then as soon as that glucose gets absorbed, the blood glucose level rises above this basal level, and that stimulates insulin release from the pancreas. And then insulin is going to stimulate these cells to take up the glucose and all these pathways that we saw before. Now what's interesting is that when we absorb the glucose into the blood and then cells take the glucose out of the blood, that increased level of glucose that comes from the meal is going to very slowly fall back down to the normal level here. As the blood glucose level falls, as the cells bring the glucose in from the blood, then that amount of glucose in the blood is going to be smaller and smaller and smaller. And as the blood glucose level gets closer and closer to this basal level, the insulin level will go down, 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 because it's recognizing that the blood glucose is fairly normal. And as insulin levels fall, glucagon again becomes the controlling hormone to maintain that 80 to 100 milligrams per deciliter. Next, let's look at the fasted state. And in the fasted state, that simply means we have not consumed a meal. If you notice, the gut is missing here, so there's no foodstuffs in the gut being absorbed into the blood and being circulated to all the tissues. So what we see is about 2 to 4 hours after a meal, it takes time for the food to get in the stomach, small intestines, large intestines. It takes time to absorb all that material. So about 2 to 4 hours, depends on the size of the meal and the content of the meal and the person. That's why we always give ranges here. But that's about enough time. So I always say about 4 hours, just to be safe. About 4 hours after a meal, then everything has been absorbed and the blood glucose level is going to be back down to the normal 80 to 100 milligrams per deciliter level. And because the blood glucose level is at the normal level, there's not an excess of glucose in the blood, and so the pancreas does not feel the need to release insulin like it was before. So what we find here is the glucose is down, it's at the basal level, insulin is down because the glucose is down, and glucagon now is going to be the controlling hormone. Glucagon is the fasting hormone that talks to the tissues and says, it is up to you to maintain what is needed to get us all through this fasted state. Now it's very, very, very important that I tell you, insulin and glucagon are two counter-regulatory hormones. There's always a little bit of insulin. There's always a little bit of glucagon. When the blood glucose goes up above the basal level, there's more insulin released, and insulin has a more dominant role. During fasting, when there's not higher than normal blood glucose, there's going to be only a little tiny bit of insulin, and glucagon is going to have the dominant role. So we're always talking about the insulin to glucagon ratio. And during the fasted state, insulin is down, glucagon is in control. Well, the whole idea of the fasting state and the glucagon is to maintain that blood glucose level at 80 to 100 milligrams per deciliter, which is more than enough glucose to meet the needs of the body during a fasted state. And the liver's job is to produce the glucose that's released into the blood to make sure that you have that level of glucose in the blood. Well, because you're fasting now, the liver is going to produce that glucose. Initially, the stored glucose in glycogen will be used as a source of glucose. Glycogen is broken down to individual glucoses. Those glucoses will be released into the blood, and that will maintain that level. As the glycogen stores start to deplete, as we run out of glycogen over time, the liver will start to synthesize glucose, and it will synthesize glucose starting with precursor molecules we talked about before. That gluconeogenesis will create new glucose that goes out in the blood. All right, that sounds pretty good. But if the liver is interested in producing glucose to be released, that's telling you that the liver is not going to use glucose as an energy source. So where is the liver going to get its energy from if it can't use glucose? That's where fat comes in. The liver will switch over to using fatty acid oxidation as the primary ATP generation mechanism to give it the energy it needs to do all this work of glucose over here. Well, where's the fat coming from? Well, we have stored fat in our adipose tissue. And adipose is going to release fats out into the blood that will circulate, and any tissue that needs fats can pick those fats up. Okay, so far so good. But who says? Well, the glucagon hormone is signaling the fasted state, and liver is going to be sensitive to the glucagon hormone. Glucagon will turn on glycogen degradation. Glucagon will turn on gluconeogenesis. Glucagon will turn on the liberation of fat out of the adipose tissue. So you see how now we've gone into the fasting mode of saying you need to take molecules out of their storage area and you need to send them in the blood so tissues can use them. Well, what is the major customer of the glucose produced by the liver? It gets out into the blood, and we talked before about brain preferring to use glucose when it can, and so the brain is going to be the primary customer, brain and nerve tissue. And then we know that red blood cell must have glucose because it can only do glycolysis, it can only do anaerobic ATP production. That's fine. Okay, now before we were talking about skeletal muscle, and we said skeletal muscle likes glucose, and it does, and cardiac muscle, but muscle is pretty good. Muscle says I can use fat. Fat's a perfectly good energy source for me. I can oxidize fats. I can get lots of ATP. So you see here now that you're going to be taking fats in by the muscle. We'll see this in a couple of slides coming up. Muscle prefers fat. So muscle doesn't require glucagon to tell it to use fat. It's just going to use that fat anyway. The difference is in this fat, in the fed state, when insulin is present, insulin will stimulate muscle to take glucose up. So use fat all the time. You'll use glucose primarily under an insulin signal. But fat is going to be muscle is going to be a big user of fat, just like liver is during the fasting times here. Okay, so these are the different processes that are going on that are happening here. And then, of course, during fast, when the liver switches over to gluconeogenesis, it needs precursor molecules so it can make that glucose. And the precursor molecules, we'll cover this later, is going to be lactate, it's going to be glycerol, and it's going to be certain amino acids. Well, where are these coming from? Well, when the adipose tissue has triglycerides, when you take those 3 fatty acids off of the glycerol molecule is leftover, it's waste, and it gets put out into the blood, but the liver will take that glycerol up and use it to make glucose. For red blood cells, they do glycolysis, and the last molecule of glycolysis is pyruvate, and since you don't have mitochondria, the pyruvate is turned into lactate. We'll talk about that coming up. And the lactate is released as waste, but that lactate gets picked up by the liver and it's used to make glucose. And the amino acids are going to be coming from the skeletal muscle. And it's important to point out here, it says protein going to amino acid. Skeletal muscle, cardiac muscle, any muscle tissue has so much protein. There's very large pools of free amino acids that support the protein synthesis. But you also have protein degradation taking place in muscle because you have old proteins being recycled and new proteins being made. And so part of that is to maintain that amino acid pool, and during times of fasting, the muscle can release amino acids into the blood, and they get picked up by the liver, and that can be used to make glucose. So during a fasted state, which we're going to say is between, right after absorption ends up to about 2 days, these are the processes that are going to be taking place. One more thing to point out here that we'll have later in the course, a lecture on the urea cycle. Whenever you use an amino acid, and you're going to catabolize that for energy, you need to pull the amino group off and you just leave the carbon skeleton. And the amino group is ammonia, and ammonia is toxic. And so we have a cycle called the urea cycle, which takes free nitrogen groups and fixes it onto the molecule urea, and that urea then carries that nitrogen safely and it gets removed from the blood by the kidney and gets released out into the urine. So during a fasted state, when the muscle is donating amino acids, typically you'll see more urea production because you're using more amino acids. This is a great concept we'll talk about. We have a whole lecture coming up about the maintenance of blood glucose and how the liver is involved in that. And so what we find is immediately after meal, so here's the glucose that's in the blood right there. A little bit hard to see, but there it is. And so our normal resting level is going to be 80 to 100 milligrams per deciliter, and that's when we're fasting, and it's a pretty stable level. If we consume a glucose meal, that glucose gets into our digestive tract, it gets absorbed into our blood. The blood glucose level rises significantly above the basal level. It goes higher than 100 milligrams per deciliter. And as that blood glucose level rises, the pancreas will sense that increased blood glucose, and the pancreas will release insulin to travel in the blood to signal to all the tissues that blood glucose is higher than normal, and the insulin will say, take that glucose up, use that glucose, let's get that glucose and put it to use. Well, if that's the case, when insulin comes out because blood glucose is elevated, glucagon now says, wait a minute, we're not fasting anymore because we have glucose available, so I don't need to be quite so active, and you see a slight decrease in the glucagon level here. So when insulin comes out to play, glucagon kind of goes away. That's what I say. But you see it doesn't go to zero, but you see less glucagon when you have more insulin. Okay, so as the insulin comes up in response to the blood glucose, that's going to stimulate glucose uptake by the cells, and so the glucose level will start falling, and it comes back down to the normal basal level. And as the blood glucose level comes down, so does the insulin level. It'll come down, and eventually as the insulin level comes back down to normal, the glucagon will start to rise back up again. Okay, so that's what's going on after a glucose meal with insulin and with glucagon. But what happens after this? What happens when you're at this point down here? Well, this graph is showing you when you've had a glucose meal and your glucose is coming in from the absorption from the gut, the glucose is being used as energy. It's going to be that ingested glucose. But very quickly, over a period of 2 to 4 hours, that glucose is absorbed, that glucose is taken into cells, and your blood glucose level is going to fall off, and the glucose being used from the diet is going to go down. But that's where the liver kicks in, and the liver says, wait a minute, my job is to maintain blood glucose at 80 to 100 milligrams per deciliter. And I am recognizing that the supply of glucose is falling off. And that's when it will say, I have stored glucose in the molecule glycogen. And the liver will say, I better start hydrolyzing my glycogen so that I can be producing free glucose units for the blood. And so you see at a period of about 4 hours to 8 hours, you see a rise in glycogenolysis, so the liver can make sure to be producing enough glucose for the blood to maintain that 80 to 100 milligrams per deciliter. But we know that glycogen is a polymer. There's a fixed amount of it. It was synthesized. It was made. And as you use that polymer up, then you're going to start to run out of glycogen. And so you reach a peak at about 8 hours, but then the molecule's getting smaller and smaller and smaller. There's less and less and less glucose there. So the amount of glucose coming off of glycogen is going to fall away, and it's going to tail off here. So over 8, 12, whatever, 16 hours or so, you reach a peak early on, and then it just very slowly falls away. But then the liver says, okay, I recognize that my glycogen is running out, but I also know that my job is to maintain blood glucose at 80 to 100 milligrams per deciliter, so how can I keep glucose production going? That's when gluconeogenesis starts. You see, when you reach that peak of glycogenolysis and you start to come down, the liver will bring gluconeogenesis activity up so that will increase as the glycogen degradation starts to fall off. And by doing that, you can maintain that very steady blood glucose level. Okay. Now, this is what insulin is doing in response to dietary glucose. The blood glucose level has gone higher than 100, and so insulin comes out to help the cells take up that glucose and bring the blood glucose back down to the normal level. This is what's happening under the presence of glucagon. When the insulin has gone back down, the blood glucose is normal. Glucagon is working on the glycogen breakdown pathways and gluconeogenesis to stimulate these pathways so that you're making sure to send glucose out into the blood by either of those pathways. So this is just the words here. It's saying exactly what I said before. The goal is to maintain that blood glucose level, and you've got glycogen stores and you've got gluconeogenesis that can do that. So this really is just the words. And then the adipose tissue during fasting, as you saw, you have the liver that needs the supply of energy and it's going to use fat, and you have muscle, which needs a lot of energy and it can use fat. And so the adipose is going to be stimulated by glucagon to hydrolyze the stored fat, the triglyceride, and make free fats and send those out into the blood so that other tissues can pick these up. Okay, so during the short fast, you see that the liver's in charge of blood glucose, and adipose is in charge of making sure there's fat for everybody. I didn't mention the ketone bodies yet, but that'll come up a little bit here. When the liver is using fatty acids as an energy source, those fats produce a tremendous amount of ATP very, very quickly. And so the liver can achieve energy sufficiency very, very easily. And the liver is pretty smart. It says, I've got all this extra energy and I don't want to lose it. So it takes the products of fatty acid metabolism, the excess that it doesn't need, and it's going to make these things called ketone bodies. They're just small ketone molecules. And they get released into the blood and they'll circulate. And any tissue that has mitochondria can take those ketone bodies up, turn them back into the products of, ready for ATP generation and use them to make ATP. We'll have a lecture on the ketone bodies coming up as well. So during a short fast, the brain says, well, I want to use glucose and I'm happy with glucose and that's all I'm going to use. And the red blood cell says glucose is all I can do. I've got to have glucose. I need my glucose. But muscle says, you know what? I need my energy. There's not a lot of glucose. I've got fatty acids, but I sure do like ketone bodies. And so muscle will be the primary customer of the ketone bodies during a short fast here. And next we come to the long fast, which is described as the starve state. And again, our book calls that as being 2 days of fasting. Some books will say 3 or 4. It kind of depends. These are ranges. It really is defined by the metabolic state of the body. But 2 to 3 days. I'll go with 2 because our book does here. Okay, so here's the thing that's telling you the story. When we look at fasting over a period of days, and days of starvation here, we're looking at time 0 here, before really you've entered into that long fast, that starving state here, there is glucose in the blood because the liver is producing glucose by first glycogen and then gluconeogenesis. And we know that the adipose tissue is producing free fat, which is giving fats to the liver, so the liver has its energy sources. So you will see fatty acids in the blood and you will see glucose in the blood. But as your time of the starve state goes forward, like 1 day, 2 days, 3 days, 4 days, the liver says, well, my glycogen is long since depleted, and the only way I can produce glucose is through gluconeogenesis. And if you remember from gluconeogenesis, you're making glucose from starting materials. And one of those starting materials is the muscle, and the muscle is donating amino acids. And the muscle says, like for an overnight fast or 24 hours or even 48 hours, the muscle says, I have more than enough amino acids. I can share what I have. I'm not too worried about it. But after about 2 days, during the normal fasted state, the muscle starts to notice that its supply of amino acids is depleting. And it recognizes that if it keeps giving off amino acids, it will no longer have enough amino acids to meet its own protein synthesis needs. Now, just because you fast for 2 days or 3 days or 4 days, your body survives. Your processes keep going on. You breathe, your heart beats, your muscles function, everything is fine. And so the muscle just says, I need to be more careful. I can't give away my amino acids. And that is a major source of a precursor molecule for gluconeogenesis. So the liver says, I've got to do 2 things. I've got to find a new source of material to allow me to synthesize glucose, and it would be helpful if the body would use less and less glucose. That would be very, very helpful. Well, if you recall, the liver is using fatty acids as an energy source during fasting. And of course, the fatty acid is more than sufficient to meet the energy needs for the liver. And there is excess energy. And that excess energy is used to make these things called ketone bodies. And after 6 hours, 12 hours, 18 hours, 24 hours, the ketone body level in the blood gets higher and higher and higher and higher. And after about 2 days, maybe 3 days, the level of ketone bodies in the blood is now high enough that the transporters at the blood-brain barrier recognize those ketone bodies, and they will start to transport those ketone bodies in. This is only in the starved state now. 2 days out is when you see appreciable ketone bodies being imported. And those ketone bodies will go into the brain tissue, into the mitochondria, and they will be used to support oxidative ATP production. And there is plenty of energy for the brain. It's very, very satisfied for its energy. And it says, you know, I don't need to use so much glucose anymore because I have these ketone bodies. And since brain is really a major customer for the glucose being produced by the liver, then as the demand for glucose goes down, the liver says, great, I don't have to make as much glucose anymore because the brain doesn't need so much glucose. It's not zero. There's still a little bit of glucose going in, but the main source of energy is going to be those ketone bodies. Now, red blood cells cannot use ketone bodies because they don't have mitochondria. But they will use glucose. You see that solid arrow there. They are going to become the major customer. And so red blood cells making lactate, that lactate will still be a precursor for gluconeogenesis. And over here, the adipose tissue is still breaking down triglyceride to free fats, and the glycerol is still being produced, and that's going to be a precursor for gluconeogenesis. And so the liver has what it needs here. So what we find is, as the fasting period, the starvation period progresses, the glucose production will come down to a lower level, which the liver can maintain over time. And the reason that it's coming down is because the ketone bodies are going up. And there's that 2-day crossover. You know, between 0 and 2 days, the ketone bodies, they stay, low enough. Muscle, skeletal muscle, cardiac muscle can use it and stuff, but certainly brain is not really going to be a major customer. But after a couple of days here, the level gets high enough and the production gets great enough that the brain can start using those. And that's what makes all the difference in the world. Here are the ketone bodies over here, and we have a lecture on those coming up. We'll talk about them. So here's the words again about brain and liver, and you can look at these and you can hear, what I was saying before. The main thing is, since the muscle is no longer giving quite as much amino acid, you're not going to be taking the amine groups off of those amino acids, and so the need to make urea is not as great, and so blood urea nitrogen, or the urea production, is going to decrease as well. Again, we're going to develop all of these in very, very good detail in upcoming lectures. And of course, the adipose tissue being the source of fat, as you can see. Now the question is, how long can we fast? And that's where it comes up to. Well, you see how this works. As long as the body has fat, as long as adipose has a store of fat, that adipose can be used to produce fats. Those fats can go to the liver. The liver can get the energy it needs to make ketone bodies to keep the brain happy. As long as you're using those fats, you're making glycerol, and that glycerol goes to the liver, and that glycerol can be used to make glucose, and the red blood cells can be happy. It's when we run out of fat, that's the problem. When a person's fat reserves are pretty well gone, then you have no choice but to go back to amino acids, and you see muscle wasting and loss of muscle tissue there. That's one of the biggest problems. Okay, so this is a big deal, this introduction lecture, and I always know that the students just get bamboozled by this lecture, especially if you haven't done metabolism before. But the point of this lecture is to give you the overall big picture of metabolism, tell you some terms to learn, and you can start getting an idea of the logic of metabolism, why tissues act the way they do, and what happens. We're going to go over this again and again and again and again, and it's going to make so much sense as we build each piece of this puzzle and put it together. So for now, just enjoy the ride, learn the terms, and we'll progress with the next. Thank you.